Okay, so I'm going to talk to you now. We've introduced the notion of network topology. I'm going to continue talking about, I'm going to talk about what simple things we can calculate from networks. So what are the main quantities that we can calculate? Well, first thing we can do, it's obvious, we have a little baby network here. We have a node. We can count how many things are connected to it. In network language, we call this the degree of the node. So this node has a degree of 5. Now, if we have a directed network, it gets a little more complicated. For instance, if this is a transcription factor, it's regulating things. We have the notion of what's called in degree and out degree. So this one is regulated by 3 things, as an in degree of 3. It has an out degree of 5. Okay, and in any case, we can describe this more often in terms of transcription factors. Then there's another notion that's a little bit harder to immediately grasp, called clustering coefficient. And basically this says for a given group of nodes, how connected are they relative to being completely connected, okay? So if we, let's say, take a look at these 6 nodes here, and let's say that we have these 2 connections, we have these existing filled in possible connections. We can ask how many connections could we have in addition. So for instance, we could also have a connection from, say, C to D in addition to the existing connection from A to C, and we can compute essentially the ratio of those 2 numbers, and that gives us the clustering coefficient. There's a more complicated formula, but that's the intuition. It's the number of connections as a fraction of the total number of possible connections. Oh, actually, I have the formula here. This is the formula for the clustering coefficient. And you can, of course, look at it for each neighborhood around a given node. So another quantity that we often calculate is the quantity called path length. And pretty obvious. So it's the number of edges along a path. So, if we traverse through this thing, we have 3 different edges. And, the meaning in, say, a transcriptional regulatory network would be how many transcription factors do you have to go to until you reach a final target? How far away is this pink TF from its final regulation? And then, of course, if we have a network, here's a little baby network here, we can calculate the shortest path in the network to travel from any one vertex to another. So if we call this vertex I and this vertex J, we could go this way, this way, this way. And obviously the shortest path in this baby network is 2 hops. So that becomes the shortest path length. So that's between I and J. And then, of course, we can average this across all different I's and J's in the network. We can get the characteristic path length or the average path length in the network. And networks that have a small value of this characteristic path length are said to have a small world property. So if you imagine if you have a network where everything is connected to everything else, obviously it's going to be a small world. It's going to have a shortest path of 1. But as we knock off connections, we don't have complete connections. If it still has an average number of hops fairly low, we'd say it's a small world. And this is very related to kind of a social science type of concept where, people say, well, if you take 2 random people, how many hops do we need to get from A to B? If we had a very spread out network, you need a lot of hops. And it turns out our worlds are actually more small worlds, as we'll talk about in a little bit. And there's a characteristically low average path length. Then there's the notion of network motifs. And that is, if we take a big network and we look at a subpart of it, we can often see characteristic patterns. So for instance, here, this is a transcription factor regulating 2 targets. People use the term signal input module, SIM, for this, okay? You could have a MIM, where these 2 targets are regulated by 2 things. We also have the notion maybe that you've heard, like a feedback loop. So here, this regulates that regulates this, but then this last thing goes back and regulates the first thing. So there's some notion of a feedback here. There's also the notion of a feed forward connection. So let's think about how that works. This thing would regulate something else. It regulates something else, but this also regulates the final target too. right? So, there's what's called a feed. This is called feed-forward construction. This is sometimes associated in the network science literature with a kind of like sort of buffering. So we have this thing is being regulated by two things. It's really being regulated by this pink thing, but this yellow thing kind of buffers it a little bit. And then if we look at a more complicated network diagram, we can decompose it into these submotifs. So if we look at this little bit of hairball here, we can see how these motifs can potentially decompose it. So for instance, let's see what's going on. So this SBF transcription factor directly regulates, say, this POG1, but notice it's also involved in feed forward loop too. You see with this other thing, and then there's a number of other feed forward loops in this picture too. And if we have a very complicated hairball network, one way of understanding it is to look at the prevalence of all these little network motifs. So here's many different network motifs. For instance, that one contains a transcription factor, another contains a microRNA, another contains a target gene, and here's the direction of regulation. For instance, this is a feed-forward loop involving a microRNA, a transcription factor, and that regulates another transcription factor. And we can count in our network how many times we see this, and that that we often find some network motifs are overrepresented in our network, and that gives us some understanding of how the network works. Another concept in the network is a clique. We're very used to this concept in a social context. This is a group of people that talk to each other all the time. They're all connected with each other, and often we want to take a bigger network and find a small part of it that's completely connected, or a clique. There's also the notion of a defective clique, whatever that means, where it's almost completely connected, but say we're missing one connection. All these people here talk to each other constantly, but P doesn't directly talk to Q, whatever that means.